Hi everyone, welcome to the Basic Science series by Dr. Lokinder Kumar. I have created this program to promote scientific knowledge among students and young researchers. In this presentation, I will guide you to a step-by-step process of gene optimization, taking an example of Pseudomonas aeruginosa LasR protein. In this episode, you will know how to find a gene sequence of a particular protein, how to check that if there is a rare codon present in the gene sequence, and how to remove and optimize the codon to express your protein of interest. You can use this as a guide to optimize your gene of interest. Before starting this presentation, please watch my previous presentation on the basic concepts of gene optimization. That episode will explain about the need of gene optimization during protein purification. That presentation explains in detail about the concepts of rare codon in a gene sequence. Alright, let's start with LasR protein. This is one of the protein produced by Pseudomonas aeruginosa. It is a regulatory protein which is used in cell-to-cell signaling in Pseudomonas aeruginosa. Since we are taking LasR as an example here, so I will not go in detail about the LasR protein structure and function. Coming on to the first part, that is how to find a gene sequence. To find a gene sequence, type the NCBI web address which is mentioned on the slide on your Google search window. What you are seeing on a computer is the snapshot of NCBI website. Then go to the red arrow and click on the Gene tab which is present on the right side of your slide. Now, after clicking the Gene window, what you are seeing is a snapshot of Gene window of NCBI. Then type LasR in the search box of NCBI-gene window which is highlighted by a red arrow. After clicking Search tab, what you will see is many results for LasR protein. Then choose on the basis of particular strain that you are working with. For example, I am working with PA1 strain, so I will choose LasR protein of PA1 strain, which has the ID number 881789. ID number is very important, so always note down the ID number of your gene. After clicking the LasR tab, you should see the window showing the details and position of LasR gene. The details of gene are shown in blue box. Now click on the FASTA tab shown in red arrow. After clicking the FASTA tab, the sequence of gene will pop up on your computer screen. The gene sequence is in blue box. Copy the whole sequence of that particular gene. Very importantly, look for the start codon at the start of the sequence which is ATG and stop codon at the end of the sequence which is TGA. This gives you a fair idea that you have a complete sequence of your gene, but you should reconfirm with the published paper. Usually, supplementary sheets of those published paper have the sequence data for the gene. Now, next step is to check the rare codons in the sequence. Go to the codon checker tool and the link address is shown on your computer screen. After opening the address, you should see the codon checker window. Now, type the name of gene in the white box shown by the red arrow. Paste the sequence of LasR gene in the second window which is shown by a blue arrow and click Analyze Sequence which is mentioned by black arrow. Results will show you the number of bases in the gene, and in this case they are 720. It will also show you number of codons, which is in this case 240. Further, it will show a table showing the list of rare codons for particular amino acid. See the table, it shows that this sequence has 4 rare codons for arginine and 2 rare codons for glycine and so on. These codons can terminate the translation process, therefore we need to optimize the sequence according to the E.coli codon preferences. To perform codon optimization, we will use JCAT codon adaptation tool. There are other tools also, but I have used this tool in the past and got good results with protein expression. Paste LasR gene sequence in the box shown by red arrow. Now select E.coli K12 by clicking on eukaryote which is mentioned by blue arrow. Now perform the codon optimization and you will see the results in the form of chart showing the fluctuation relative adaptiveness in unoptimized DNA, and in case of optimized DNA the relative adaptiveness will be very smooth. It will also show you the CAI value which is codon adaptation index value for the unoptimized sequence. In our case, the value is 0.3 for unoptimized, and after optimization, the value is 1. For an optimized DNA, the ideal value of CAI is 1. It will also show you improved DNA sequence. Copy the sequence in a new file. Now, to recheck the improved DNA, you can perform rare codon checker exercise again and check whether you have any rare codons left. In this case, we have performed the checking, and as you can see, there are no rare codons. Now you're ready to send your sequence to gene synthesis companies, and the services will provide you your gene cloned in a desired plasmid vector. Congratulations, now your gene is fully optimized. I hope you understand the step-by-step process of gene optimization. In the next episode, we will discuss more about new techniques that are used in basic sciences, so please stay tuned and enjoy the presentations. If you haven't subscribed the channel, please subscribe the YouTube channel and share the presentation with students and young researchers. Please forgive my mistakes. Thank you and namaste.